The Genuine Article. Discussion and commentary based on articles from JACC.
Hello, I'm Tony DeMaria, the editor of JACC, and I'm here with Dr. Rakesh Kukreja and Dr. Li Shi. And Dr. Kukreja is the director of the Heart Center at Virginia Commonwealth University, where Dr. Shi is an assistant professor, and they've authored a fascinating study about to be published in JACC that deals with the potential of nitrate-nitrite supplementation to perhaps eliminate or at least reduce the toxicity from doxorubicin. And as we all know, doxorubicin is a very valuable chemotherapeutic agent, but it's limited in large measure by its cardiac toxicity. So our authors hypothesized that by giving nitrate-nitrite supplementation, they might increase NO, nitric oxide, and in that way limit the toxicity. And they looked at a group of mice in whom this was tested, where they measured pressures and looked at necrosis, apoptosis, and peroxidation, and showed, in fact, there were favorable changes in all of these parameters by the nitrate-nitrite administration. Congratulations, gentlemen. A very nice study. First of all, clarify for me, if you will, the difference between the NOS-dependent and independent pathways of nitric oxide.
The difference between the two pathways is that nitric oxide independent pathway does not use nitric oxide synthase enzyme for the production of nitric oxide in vivo, whereas the nitric oxide dependent pathway uses L-arginine as substrate for production of nitric oxide.
So by giving the oral nitrate-nitrites, one could be able to use this independent pathway, and I understand that there's conversion to nitric oxide in the gut. Can you tell us about that?
Yes, yes, in the oral cavity, there are very rich colonies of bacteria. These bacteria used to convert nitrate into nitrite, so eventually convert into the NO. So it is very important phenomenon.
So tell me, you did show beneficial effects of the nitrates in these mice. How much nitrate would have to be given, and does it have to be given orally, or can it be given intravenously?
We actually add 1 gram of sodium nitrate into 1 liter of the drinking water, give to the mice 1 week before and after doxorubicin treatment. So we subsequently, because of your JACC reviewers' request, we did also subcutaneously infusion of a similar concentration of nitrate into the mouse body. We found no protection to the doxorubicin cardiotoxicity. So what we believe is the oral ingestion is the only way to show the nitrate-induced protection.
Well, what would this convert to in humans? How might humans increase their nitrate ingestion?
And we actually studied the FDA the conversion for mice and human dose. So we think the dose we are using is highly clinically relevant. It's only about 400% of the WHO acceptable daily intake, which is called ADI, WHO ADI.
Now, one of the things we have to acknowledge is that the magnitude of the effect of the nitrates was relatively modest. Did you do any dose ranging studies?
We didn't. We mostly translated the dose previously used by other investigators in the ischemia reperfusion model, they also show the similar dose of nitrate actually protect ischemia reperfusion injury. This was published in the Proceeding of the National Academy of Science back in 2007.
Now, based on these data, do you think that someone will take this forward to do a clinical trial as to whether nitrate supplementation in a clinical setting would offset the doxorubicin toxicity?
Absolutely.
Absolutely. This is because nitrate is relatively inexpensive inorganic ion, and also it is rich in many of the fruits and vegetables, particularly leafy ones. So there could be two approaches, whether one can use the nutrition approach, and the other could be the use of dietary nitrate supplementation with inorganic ions.
Well, excellent work, very interesting. Clearly this would be a very, very valuable clinical advance to be able to reduce the toxicity of doxorubicin. Thanks very much for being with us. For Inside JACC, I'm Tony DeMaria.